You're welcome to this session in the series of antimicrobial susceptibility testing, what we call antimicrobial susceptibility testing, which we are looking in this session. We are looking at. We want to see which methods we use, and under susceptibility testing, we do what we call dilution susceptibility testing, where we are looking at MIC and MBC. So here we shall be looking at MIC and MBC. Then another one we shall see at modified Kirby-Bauer method, which is the one commonly used in the laboratory. Then we shall end with Stokes method, which is rarely used, and for it uses both the control and the test at the same time, which is different from Kirby-Bauer method. So under dilution susceptibility testing, and as we are beginning, we always remind you to subscribe, like the video, and share the comments. So we want to see how do we determine minimum inhibitory concentration and minimal bactericidal concentration. And among the requirements we need is the broth, or we need the broth or agar. So after adding the broth within the tubes, we make serial dilution of the antimicrobial agent, which we want to determine its MIC and MBC. So if it is chloramphenicol, we put chloramphenicol. If it is erythromycin, we use erythromycin. So we use the antimicrobial agent that is in equation, whom we want to determine the minimal inhibitory concentration and minimal bactericidal concentration. So after having these two requirements, what we do is we make serial dilutions. We make serial dilutions using antimicrobial agent and the broth. After making serial dilutions, beginning with the strongest, this one is the strongest, or with the highest concentration, and this one will be the one with lowest concentration. So we are using 7 tubes, and in these 7 tubes we are making serial dilution, beginning with the one with 64 concentration to 32, 16, 18. We keep reducing until we go to 1. And even more, we can even move up to more. So after making these serial dilutions of the antimicrobial agent, remember we were diluting this agent using the broth or the agar, so it can support the growth of microorganisms. So after making the serial dilution, which is point number 1, point number 2 is we add the standard inoculant. We add, or we inoculate a standard inoculum, which has been measured according to 0.5 McFarland standard. So we come and add the inoculum, the microorganism. We add the microorganism in each tube, the same amount of the microorganism in question, which we want to test its susceptibility against this known antimicrobial agent. So after adding the same standard inoculum, or the same amount of the standard inoculum microorganism, we intubate. Number 3 is we shall intubate overnight. Intubate the tubes overnight, and the maximum we normally intubate 12 to 24 hours after intubation, and we do it at the temperatures of 35 to 37 degrees centigrade. So after intubating overnight at this temperature, we come and we read. We obtain the tubes and we check for here number 4 is after overnight intubation, we check for the tube with visible growth. And we check the ones without visible growth. And After inoculating and intubating at 37 for overnight, and we check in the tubes and we find tube 1 has the bacterial growth. This is the bacteria. Growth is too much. We find tube 2 also has some growth, and we find this one has also some growth. This one has much growth, and we find here there is little growth, and we find tube 4 has no visible growth. So our tube 4 becomes the MIC. So this one becomes the minimum inhibitory concentration because it is the tube with the lowest concentration, 8, with no visible growth. Here there is no, the one with the lowest concentration with no visible growth of the microorganism after intubation becomes our minimal inhibitory concentration. So after we check for the presence of visible growth and we record the MIC is the tube with lowest concentration with no visible growth. No visible growth. So after determining the MIC, which is the minimal inhibitory concentration of the drug, then we can also determine the MBC. What is minimal bactericidal concentration? Minimal bactericidal concentration is the lowest amount of the drug that can kill 100% bacteria. So this one does not depend on the rather than visible growth. So this one, it does not depend on the visible growth, but rather it looks for the lowest concentration where there is maximum killing of the bacteria. So it does not depend on only visibility, like in MIC. MIC depends on the visibility, but for minimum bactericidal concentration is the concentration after doing serial dilutions that can kill 100% microorganisms. Even it does not depend on the visibility of the growth. That is what we call minimum bactericidal concentration. So we are saying that this dilution susceptibility testing enables us to determine the MIC of the antimicrobial agent and the MBC of the antimicrobial agent, whereby we have said the MIC is the minimum inhibitory concentration, which is the lowest concentration that shows no growth of microorganisms, visible growth. Whereas MBC is the lowest concentration, which kills 100% of the microorganisms. Then the number 2 we can see in this video is Kirby-Bauer method modified. Modified Kirby-Bauer method, which is we said is commonly used. This is commonly done in the lab. Commonly used is Kirby-Bauer method. And in this method we need requirements. We always use Muller-Hinton-Egger. We use the sterile wire loop. Sterile wire loop together with the swab, sterile swab. Then we shall need the organism to be inoculated. We shall need the discs containing the antimicrobial agent. We shall need the discs, and we shall need the incubator and control strains. So among the procedure of this method, we first obtain the Muller-Hinton-Egger. If this Muller-Hinton-Egger is already prepared and is in the fridge, because after its preparation it can be kept between 2 to 8 degrees centigrade for 2 weeks. So if it is already in the fridge, we remove it and we put it at room temperature. So we leave this Muller-Hinton-Egger. If it was in the fridge, Muller Hinton agar at room temperature for like 30 minutes. Here we want it to obtain room temperature. After putting, if it is freshly prepared, we first leave it for also like 30 minutes to set very well. And after the Muller Hinton agar is ready to be used, what we shall do, we shall obtain our plate with the organism, where the organism is to be tested for sensibility, for susceptibility is, and we shall use a sterile loop, use a sterile wire loop, and we touch on 3 to 5 colonies. So you touch on the 3 to 5 colonies. These colonies should be of the same color and growth. So we normally pick from 3 to we touch using a sterile wire loop. After hitting the red hot the wire loop, then we touch 3 to 5 colonies and we inoculate them in 3 to 4 mils. We inoculate them in 3 to 4 mils of saline, of normal saline. We can use normal saline here, or we can use distilled water. So after we have put these 3 to 5 colonies, we have picked them, we have added in 3 to 4 mils. Now we come and adjust the inoculum, or we can use the word standardize. We standardize the inoculum to 0.5 McFarland standard, whereby after you have mixed this inoculum with 3 to 4 mils, you hold it in the presence of light and you keep comparing it. You compare it by adding more of the distilled water until it matches. If this is my 0.5 McFarland standard, which is it is prepared using barium sulfate, and maybe I have also my test inoculum. So I put them in presence of light and I keep comparing them by adding this until it matches to this. If it is low, I add more inoculum, more colonies, and if it is much, I add in more distilled water until it matches with the 0.5 McFarland standard in the presence of enough light. After matching it or adjusting it to McFarland standard, here we shall obtain a sterile swab. Using a sterile swab, this sterile swab, we insert it in the inoculum prepared. The sterile swab, we pick the standard inoculum. Then we remove excess. How do we remove excess? By tapping on the side of the tube. And after removing excess, here we shall strike the inoculum on Muller Hinton egg using at 60 degrees centigrade until it evenly distributed all over the Muller Hinton plate. And after we have inoculated, this is the process of inoculating. After inoculating using a sterile wire loop or using a sterile swab, even all over the Muller Hinton agar, and we normally hold the swab at an angle of 60 degrees centigrade until all of it is streaked all over to make a smooth or evenly distributed streaking. After this, we leave it for 3 to 5 minutes. We leave the Muller Hinton, the streaked Muller Hinton, for 3 to 5 minutes when covered to dry. This one is to enable it to dry. And after drying, within 30 minutes after drying, we shall get our sterile forceps. And this sterile forceps we pick the antimicrobial discs with the antimicrobial agent, and we apply them on the Muller-Hinton plate. If this is the Muller-Hinton plate, if this is my plate, and it should not be more than 6. So if I put one at the center, I put one disc at the center, I can put other more 5 around. And we say that these discs applied, 6 of them, in between them it should have a distance of at least 25 millimeters. Even here, the distance should be 25 millimeters from each other, and from the margin it should be at least 15 millimeters. Even this one from the margin should be 15 millimeters away from the margin of the plate. So this is my Muller-Hinton egg. So I have used the sterile forceps to apply these discs. These are microbial discs. 6 of them 25 millimeters apart and 15 millimeters from the edge of the plate. After doing this, applying them, we cover the Muller-Hinton agar, and we should incubate it at 35 to 37 degrees centigrade within 30 minutes. And we leave it for overnight, and we incubate it for overnight, which is always the required is 12 to 24 hours. After 24 hours of incubation, that's when we come and read the results to see if there is a zone of clearance. So after overnight incubation in aerobic, this one should be done in aerobic, that is in the presence of oxygen. So after incubation for 24 hours, we come and read for the zone of clearance. If we find maybe this one, there was zone of clearance around the disc, and we find this one, there was a small zone of clearance. This one, no clearance, and this one was also, there was a zone of clearance around. This one was resistant, or maybe this one was intermediate, and here we see no zone of clearance. Here, no zone of clearance. So we shall say, if this was disc number 1 and number 3, we shall say, among the reports, on the reporting, we shall say disc 1 and 3, they are resistant. Then we shall see disc, if this is 2, 3, this is 4, 5, and 6, we shall say disc 4 and 5, no, we shall say disc 5 is intermediate, and we shall say disc 6 and 4, they are susceptible. How do we measure the zone of clearance? Already the disc is 6 millimeters. So we get a ruler and we measure the diameter. Already this is 6 millimeters. So we measure here the zone, or we can get a ruler and we measure across by measuring the diameter. So after measuring, we compare it with the NCCL Kirby-Bauer standard. So we normally compare, to report, we normally compare with any CCLS standard chart to know if this is resistant or intermediate or susceptible. For intermediate drugs, these ones, if they are used in higher dose, they can treat the condition. But these ones are susceptible even at their recommended dose, and these ones are resistant even though given in higher doses, the microorganism cannot respond. So this is the Kirby-Bauer method, which is commonly used. And the last one is known as Stokes method. But this Stokes method, for it, is not commonly used, but we said in this method, both the test inoculum and the standard are put at the same time, are run at the same time, but not commonly used. But as before we finish this, we are going to see that there are different controls. Controls, because everything we do in the laboratory must be controlled, and the controls we normally use E. faecalis, ATCC 292-112. We can even use, E. faecalis, we can use cotrimoxazole, also is a positive control. And another thing we have to make sure is to standardize our inoculum, to make a standard using 0.5 McFarland standard. That is another control. Another control is always using molar Hinton agar from trusted sources, which is sterile. Another one is using potent discs, which are not expired. Always check the expiry date and also the storage of the discs. And before using these discs, they are supposed to be brought at room temperature before applying them. So these are some of the controls we have to always make sure we put into considerations when we are doing these susceptibility methods. And I want to end by looking at the factors that affect susceptibility testing. Lastly is to look at the factors that affect antimicrobial sensitivity testing. Factors that affect. This is commonly asked in exams. And one of the factors we can talk about incubation time. We can talk about incubation temperature can affect. We can talk about potence of the disc. We can talk about another point we can talk about is the size of the inoculum or the degree of size of the inoculum. Can talk about among the factors to consider is the distance between discs. So these are some of the factors we can talk about under this. Not forgetting about the depth of the media. Media on molar Hinton egg. So these are the factors. How does depth affect? The depth, you must tell us when you're discussing this, tell us the ideal. After telling us the ideal, you tell us in case it is above and in case it is low, what happened? And the ideal should be 4 millimeters deep. So if it is greater than 4, we are going to look to get false low. We shall get false results. We shall get false resistant results. and if it is less than 4 millimeters, we get false positive. Here we get false positive, here we get false negative when it is above. So you tell us the ideal. The distance between the discs, the ideal should be 25 millimeters. If it is greater, you tell us what happens greater than 25. And if it is less than 25, tell us. If it is greater than 25, this is the best. There is no effect. But if it is less, it can give false positive due to the drug combination when they come together during zone of clearance. So this one is which we call the zone of clearance. Then the size of the inoculum, we said it should be matched with 0.5 McFarland standard. And in case it is greater and in case it is low, what happens? If the inoculum is greater, it will lead, meaning the bacteria are high, it will lead to false positive. It will lead to false negative because the microorganisms are many, and if it is less, it will lead to false positive. That is when it is less than 0.5 McFarland standard. And about the potence of the disc, if the disc is less potent, what happens? And if it is more potent, what happens? If it is less, it will give false negative, that is which are resistant, and if it is more, it will give false positive zones of clearance, greater zones of clearance. Negative means there is no zone of clearance, then positive means there is greater zone of clearance, but which is false. Then incubation temperature, the ideal should be 35 to 37 degrees. Then tell us if it is greater than 37, what happens? And if it is less than 35, what happens? If it is greater, it will depend. But some microorganisms, it can lead to false positive. Those ones that are good at higher temperature, and those which cannot grow at higher temperatures gives false negative, and then even below, it gives false positive. Because the growth will not be. So the standard should be this. Then incubation time should be 24 hours. So if you read beyond 24 hours, greater than 24 hours, it will give false positive. And if you read less than 24 hours, it will give false negative. So it is very important to know how to discuss these factors affecting susceptibility testing. Thank you so much for being with us, for watching from the beginning until the end. We are always privileged to have you at our channel. Thank you so much. Always remember to subscribe and share your comments. Thank you so much.